In this video, we'll explore key concepts in the IB biology topic of diversity of organisms. We'll start with variation, then discuss species concepts and speciation. From there, we'll dive in deeper to the molecular level, starting with chromosomes and karyotyping. This will help us understand the genome diversity that underpins organism diversity. Finally, we'll take a look at genome sequencing that allows us to study this. By the end of this video, you should be able to explain how this variation arises and discuss some of the issues involved in studying diversity. Life on Earth owes its success to variation. Variation includes all the biological ways organisms can differ from one another across all three domains of life: archaea, bacteria, and eukaryota. These organisms are individual living entities composed of one or more cells. This can be as simple as E. coli, a single-celled bacteria, or as complex as Homo sapiens, humans. We are multicellular eukaryotes, and each individual human has different biological characteristics, for example, blood type or skin color. Let's think about how these biological traits can vary. ABO blood type falls into 4 distinct categories. You can either be type A, type B, type AB, or type O. There isn't a gradient between these classes. We can therefore describe the ABO blood groups as an example of discrete variation, where the presentations of the trait show distinct, separate forms. Other examples of discrete variation are snapdragon flower petal colors or pea seed color. What if a phenotype is not expressed in distinct categories? This is continuous variation, as seen in human skin color. Skin color can vary from light to dark because many factors come together to add a little to the phenotype. These factors are genes, regions of DNA in our cells that influence specific biological traits. As an increasing number of genes contribute to a trait, the more likely it is that the trait will show continuous variation. In contrast, ABO blood group is controlled by only one gene, so it shows discrete categories. Because genes influence our biological characteristics, variation ultimately originates in genetic variation. Our genomes are composed of sequences of DNA nucleotide bases. As this sequence changes through mutation, we develop different traits, increasing variation. This can be seen within a species, as we show with skin color, or between species. Humans and chimpanzees display different traits and behaviors, despite being very closely related evolutionarily, because we still have genetic differences. So let's talk a little bit about species. There are 2 dominant modes of conceptualizing what a species is. First, we have the morphological species concept. Morphology refers to the physical form something takes. For instance, both of these organisms are small birds. They have orange patches on their chests, small beaks, and grey feathers. On the right, we have a European robin. When Europeans came to the United States, they saw these birds on the left and noted that they shared similar observable traits. So, they called this bird the American robin. These were grouped as the same morphological species as robins. However, as scientific knowledge progressed, a new method of grouping species emerged. Biological species are grouped according to their ability to interbreed and produce fertile offspring. American robins and European robins are not able to do this. So, under the biological species definition, they are different species. Biological species still share physical characteristics, but these alone are not enough to determine them as the same species. You can see this in the difference between the common and scientific names. We know the common names, which are colloquial and can vary between different places and languages. However, scientific names are standardized through binomial nomenclature, a worldwide system of naming used to avoid confusion from language barriers or slang terms. This nomenclature is based on biological species, not morphological, and has a few rules. Binomial means 2 names, and nomenclature refers to a system of naming. Therefore, the assigned scientific name of an organism identifies its classification by indicating its genus and species. For the American robin, this is Turdus migratorius. The first word is the genus name, which is a wider group of organisms that contains multiple species. In this case, Turdus, Latin for thrush, refers to a group of songbirds. This is always capitalized. It can be shortened to its first initial and a full stop. Next comes the species name specific to this bird, Migratorius. Species are named based on characteristics, location, or perhaps a person. Migratorius comes from the Latin word migratorius, meaning to migrate. The species name isn't capitalized and is always written in full. Two other rules are that when typing, the scientific name is italicized. However, when written by hand, the genus and species are underlined separately. This highlights issues with the biological species concept. Organisms can be different species according to the morphological species, but sometimes produce fertile offspring called hybrids. For instance, tigers and lions can interbreed to produce ligers. Plants are notorious for their hybridization abilities, which we have used over the centuries in crop farming to produce crops with desired characteristics. Further, members of a species can live across wide geographical areas. This separation may lead to variation within the species as they adapt to different environmental niches. So, organisms may be of the same species, but perceived as different species. Testing this by interbreeding species is time-consuming and costly. However, these distributions are also key drivers of species divergence, called speciation. Speciation is the formation of two or more species from a common ancestor. It is currently impossible to state the accurate number for the amount of different species on Earth, but it's certainly in the millions, if not billions. All of these species arose through speciation from the last universal common ancestor. But what does this process look like? Let's consider two scenarios. In the first, we begin with a single population of goats belonging to the same species. Over time, a creek in their habitat gradually widens into a river, creating a physical barrier that prevents goats on one side from crossing to the other. This results in the original population being split into two geographically isolated groups. Although they are physically separated, the goats on both sides still belong to the same species, as no significant genetic differences or reproductive barriers have yet evolved between them. Over time, the two isolated goat populations adapt independently to their environments through natural selection. On the left side of the river, steeper terrain favors traits like longer legs and tougher hooves. These traits become more common as individuals with them survive and reproduce more. On the right side, dense vegetation favors smaller size and stronger teeth. As genetic differences accumulate, the two populations eventually become so distinct that they can no longer interbreed and produce fertile offspring. At this point, they are considered separate species. Another mechanism of speciation is temporal isolation, where populations become reproductively isolated due to differences in activity timing. For example, in a population of desert butterflies, some individuals begin shifting their activity to nighttime to avoid predation from birds that hunt during the day. These butterflies feed and reproduce at night, then hide during the day. Meanwhile, others remain active during daylight, relying on large, eye-like wing patterns to deter predators by mimicking larger animals. Because the two groups are active at different times, they no longer interact or mate. Over time, as they adapt to their distinct ecological niches, genetic differences accumulate, eventually resulting in the formation of two separate species. However, this showcases the complexity of defining species, as we discussed earlier. There is no single moment when two isolated populations suddenly become separate species. Speciation is a gradual, continuous process. During this transition, some individuals from different populations may still be able to interbreed successfully, while others cannot. This overlap makes classification challenging, as it reflects our human attempts to impose discrete categories on what is often a continuum in nature. What are some other examples you can think of where our species concepts become harder to apply? This doesn't mean that there aren't specific markers we can look at to determine that two organisms are from different species. Let's turn our focus inward to the genetic basis for variation and take a look at chromosomes. Chromosomes are found in the cells of all living organisms. In eukaryotes like us, they're composed of DNA and associated proteins. The simplest life forms, organisms in the domains bacteria and archaea, typically have a single circular chromosome. Each species has a characteristic number of chromosomes, which remains largely consistent among individuals of that species. While minor variations can occur, the overall chromosome number is generally stable and can serve as a useful feature in distinguishing one species from another. Differences in chromosome number and structure are often key indicators of evolutionary divergence between species. For example, humans have 46 chromosomes, while our closest evolutionary relatives, chimpanzees, have 48. Chromosome number is not unique to a species. For instance, the cultivated olive tree, Olea europea, also has 46 chromosomes. However, the genetic content within those chromosomes is vastly different, which is why humans and olive trees have such distinct biological characteristics. Over evolutionary time, different lineages have gained, lost, or rearranged chromosomes through various mechanisms. In particular, plants are well known for their ability to undergo whole genome duplication, resulting in polyploidy, where organisms have multiple sets of chromosomes. This process can lead to rapid increases in chromosome number and is a major driver of plant diversification. Chromosome loss can also occur in populations over time. One current hypothesis for the different number of chromosomes between humans and chimpanzees is that during speciation, the population that would become humans had a fusion event, whereby two chromosomes merged into one. Let's take this as an opportunity to talk about how modern science develops hypotheses and scientific theories. In science, a theory is a well-established explanation for a natural phenomenon that has a wealth of empirical evidence, and so has been accepted by the scientific community. Theories are built on hypotheses, testable statements or predictions based on existing knowledge, designed to be investigated through experiments. For instance, a hypothesis could be that human chromosome 2 is a result of a historical fusion event where chromosomes 12 and 13 from a common primate ancestor joined end to end. This only occurred in the human lineage, so chimpanzees still retain the chromosomes 12 and 13, giving them one extra pair of chromosomes than humans. This hypothesis is testable and has evidence suggesting its validity. The DNA in the middle of chromosome 2 in humans contains degenerated telomeric DNA. Telomeres are segments of DNA that are located at the ends of eukaryotic chromosomes to protect them from decaying over time. The presence of this DNA in the middle of chromosome 2 points towards an end-to-end fusion of two chromosomes. Not only that, but the DNA contents of human chromosome 2 matches the contents of chimpanzee chromosomes 12 and 13 specifically. This similarity is not seen in the DNA of gorillas or orangutans, showing that this divergence occurred later, after gorillas and orangutans diverged from the human-chimpanzee lineage, and that it's particularly relevant to the divergence of humans from chimpanzees. Since this fusion event leaves the proto-human population with fewer chromosomes than the proto-chimpanzee population, it would be a distinct reproductive barrier between the two. One key aspect of interbreeding is chromosome compatibility. This lack of compatibility would hasten the speciation process, creating a genetic barrier rather than a geographical or temporal one. So, this ape population split into two species, which over time evolved into two genera: Homo, of which we Homo sapiens are the last extant species, and Pan, which contains chimpanzees and bonobos. The evidence for this fusion was gathered using a scientific technique called karyotyping. This is when a complete set of chromosomes from an organism or cell is extracted and arranged in pairs according to length, position of centromere, and banding pattern, producing a karyogram like we see here. This is a karyogram from a human cell with the 46 chromosomes lined up decreasing in length. You can see that the banding patterns of light and dark on the chromosomes are the same in each pair. This is because homologous chromosomes have the same genes, but not necessarily identical DNA sequences. This banding reflects similar structure, not perfectly similar identity. These 22 chromosomes are autosomes, which are not sex chromosomes. You can see that at chromosome 18 there are 3 copies of the chromosome, called trisomy. This is an example within a species where individuals can have a different number of chromosomes to each other despite a consistent species chromosome number. In this karyogram, the sex chromosomes are also visible, shown here as X and Y chromosomes, which identifies this individual as a biological male. Now that we've talked about chromosomes, let's zoom in even further to the DNA sequences contained within them. Earlier, we mentioned genes. Genes contain genetic information because they give instructions to the cell to synthesize polypeptides or functional RNA. The DNA is used as a template to produce a strand of RNA, and sometimes that RNA is used as a template to produce a polypeptide to build proteins. This is the foundation of biological traits. Variation in ABO blood group is a result of variation in an enzyme protein. You can think of this like a code. Instead of using language, DNA uses specific sequences of nucleotide bases, adenine, thymine, guanine, and cytosine, to tell the cell what to synthesize. So, genes are also called coding sequences. Most of the DNA in the genome is non-coding and doesn't produce polypeptides, but it does serve other important functions like regulation and protection. Genetic variation stems from differences in this code. Genes can exist in different forms called alleles, which are variations in the base sequence of the gene. These alleles can change the expression of the trait controlled by the gene. For instance, coming back to our ABO gene, there are 3 main alleles seen in the human population: A, B, and O. The combination of these alleles determines the blood group. For example, a person with 2 A alleles will be blood group A, as their red blood cells will contain A antigens. These alleles arise through a process called mutation, where nucleotide bases in a sequence are changed from their original base, like adenine being changed to cytosine. This changes the code and can change the final product. One important kind of mutation is single nucleotide polymorphisms, or SNPs for short. Single nucleotide means that only one nucleotide base is changed in a sequence. Polymorphism means multiple forms. So, SNPs are sites where there are multiple alleles of a gene that differ by one nucleotide base, seen in at least 1% of the population. 75% of people may have the original adenine, while 25% have the mutated cytosine. SNPs can occur through several mechanisms of mutation, like errors in DNA replication or exposure to agents that cause mutation, like UV rays. Because they involve switching single nucleotides, SNPs don't change the size of the genome. This instead occurs through insertion and deletion mutations, where nucleotides are added or removed from the DNA sequence. The size of a genome is described using the number of base pairs. For example, the bacterial genome of E. coli is usually around 5 million base pairs. In contrast, the human genome is approximately 3 billion base pairs. We use the metric system to condense these numbers, as you can see here. It's important to understand that larger genomes don't necessarily increase the complexity of the organism. Grasshoppers have a larger genome than humans, and the plant polyploidy we mentioned earlier has led to Paris japonica, a lily, currently having the largest known genome in the world at 150 billion base pairs. Furthermore, in species with large genomes, the vast majority of the DNA is made of non-coding regions. So while only 13% of E. coli's genome contains non-coding DNA, 99% of the human genome is non-coding. This means increasing genome size doesn't inherently lead to an increase in the number of genes. We know all of this information about genomes because of the incredible advancements made in genetic sequencing technology over the past 3 decades. In 1990, the Human Genome Project began, with the goal to sequence the entire human genome. This means that a team of scientists set out to identify the order of nucleotide bases across each chromosome. By 2003, it had drafted the first version of the sequence, using the old Sanger sequencing method to cover approximately 90% of the genome. This type of sequencing is slow but accurate. It determines the sequence of a length of DNA by consecutively identifying the bases, making it ideal for short sequences. In the years following, new sequencing methods were developed and called next-generation sequencing. This takes the opposite approach to the one-by-one method of Sanger sequencing and produces a massive library of small, overlapping fragments from a large length of DNA. These fragments can then be rapidly sequenced and reassembled using the overlapping ends, and this is especially useful when compared to a draft template of the full sequence. This type of sequencing is less accurate than Sanger sequencing, but much faster and cheaper, leading it to be ideal for large segments of DNA. If you get your genome sequenced today, it's highly likely to be done through this method, because it can be compared to the updated human genome sequence. This ability to sequence and compare genomes, particularly human ones, has led to changes in the way we practice medicine, conduct research, and study genealogy. For example, personalized medicine involves sequencing a patient's genes and using that knowledge to tailor treatments to their specific genetics. This might involve identifying mutations associated with cancer or assessing how effective medications may be based on your alleles. In research, DNA sequencing is used in phylogeny and evolutionary biology to classify species and determine the genetic evolution of different genes or organisms. Drug research can now obtain sequences for the pathogens responsible for infectious diseases, and agricultural science can sequence crop genomes to introduce targeted genetic modifications. In fact, the entire field of genetics has benefited enormously from these new technologies, leading to increased study of not only DNA, but RNA and epigenetics. Finally, genealogists can use the comparison of human DNA sequences to trace individual relationships and population migrations. For example, paternity can be established through sequence similarities between two people. SNPs can be more or less common in different populations, and used as a marker to understand the movement of humans out of Africa. DNA sequencing technology has revolutionized large swaths of medical, scientific, and cultural research. However, this doesn't come without risks. There are ethical concerns regarding the use of DNA sequencing and the protection of genomic data. Since your DNA is unique to you, it can be used as a specific identifier, leading to privacy issues. If a company sequences your data, they could then give it to law enforcement or insurance companies, risking your privacy and safety. Since this is such a new development, government regulations across the world haven't caught up yet, and it can be challenging to write legislation on complicated scientific data and technologies that are still developing. Furthermore, there are always disadvantages to using genetic data in any given field. Identifying genetic risk factors in medicine has an emotional cost, particularly if nothing can be done about that risk. While genetic counselors may be able to compartmentalize bad news or contextualize risk levels, patients are most often going to be frightened or upset, regardless. When obtaining genomes for research, informed consent is not always given. The most famous example being the cells stolen from Henrietta Lacks in the 50s, a black woman who passed away from cervical cancer. Her cancer cells went on to become the HeLa cell line, foundational to cancer and genetic research, but her family is still fighting for compensation. This is the importance of understanding this topic. You can pause the video to review the main ideas. Hopefully, you can appreciate that the success of life on Earth all comes down to tiny little variations in our DNA, stacked, one on top of each other, over 4.5 billion years.